Good afternoon, everyone. I'm here today to talk about the method divided by our team with the title Feature Selection of Single-Cell Transcriptomic Data Predicting Cell Positions. Okay, we know the goal of this dream challenge is to select a subsize of 60, 40, and 20 gene sites, which contains the most spatial information to predict cell positions in Drosophila embryo. So our method can be roughly divided into 3 steps: feature selection, mapping, and evaluation. For feature selection step, we use 2 methods. One is called the maximum distance method, and the other one is called the genetic algorithm. For the spatial location mapping, we just use the DistMap method. And we evaluate the performance of our method by these 2 scores. One is a distance score, and the other one is a gene pattern score. Now let's look at the workflow of our method. The first step is feature selection. For sub-challenge 1 and 2, we use the maximum distance method. This method is based on Euclidean distance, which means we will calculate, for each gene, we will calculate the distance from this gene to all the other genes. For sub-challenge 3, we use a different strategy, which is a genetic algorithm. This is a powerful tool to solve optimization problem. After we got the select gene site, we will map the cells to the positions. Here we adopt a DistMap method, but rather than we just select the top 10 position directly, here we select the top 30 positions with the largest value of Matthews correlation coefficient. Then based on these 30 positions, we will find outliers from these positions. And then we will delete outlier. The rest of positions, we will select the top 10 ranked positions, also based on the Matthews correlation coefficient. And then we evaluate our method by using these 2 scores. Distance score is the score to evaluate distance from the positions we predicted and the positions predicted by DistMap method. And the gene pattern score is a correlation between the select gene expression of single-cell RNA-seq data and in situ data. Okay, now let's look at the feature selection step for our sub-challenge 1 and 2. Here we want to select a most informative and spatial gene site to do prediction. So here we just want to find a gene site that has a maximum Euclidean distance between each other. So we extract 84 gene expression data from the normalized version of single-cell RNA-seq data. So first step, we just choose 80% of cells randomly. So which means that there are totally about 1,200 cells in the single-cell RNA-seq data. We will just choose about 1,000 cells each time to avoid overfitting. Based on this data, for each gene, we will calculate the summation from this gene to all the other genes. So for each gene, we will have a distance value. We will delete the gene which has the smallest distance value, and this gene will be ranked as 84 because we totally have 84 genes. And next we delete another gene, and the second gene being deleted will be ranked as 83. By using this step, we will get a final ranking for the 84 genes. And then we just repeat this procedure about 10,000 times, and we will get 10,000 ranking lists, and we will take the average of these rankings as our final ranking. So based on this ranking, we can just choose the top 40 or top 60 as the gene site. Okay, To visualize the feature selection step of our maximum distance method, here I just use the first 3 PCs to represent each gene expression value. So here, each blue point represents a gene. So we totally have 84 given genes. We want to select 60 from them. So by using maximum distance method, we select, here the red points represent the genes we selected, and the blue ones are the genes we didn't use. Same thing for 40 select genes. From these figures, we can see the select genes are scattered over all 3D space, and they are far from each other, which we may think they contain small spatial information. Okay, next, based on the select gene site, we map the cells to the positions by using this map method. But rather than we select the top 10 positions directly, we will select top 30 positions, which means the 30 positions has a maximum value of Matthews correlation coefficients. Based on these 30 positions, that means 30 bins. For each bin, we can calculate a distance. Same thing from this bin to all the other bins, according to their spatial space. So we will delete the bin which are outliers. So based on the rest bins, we will select the top 10 bins with a maximum value of Matthews correlation coefficient. Okay, here is a step for sub-challenge 1 and 2. So you can see we use the same feature selection method and mapping method. But when we consider to select a small gene site, like in sub-challenge 3, there are only 20 genes we need to select. So we may consider to use a different strategy. Here we just use a genetic algorithm to do feature selection in sub-challenge 3. Dr. Shu will talk about that. Thank you.
So, as mentioned before, so the third sub-challenge is the most difficult one because we're only able to use 20 genes in this setting to predict the spatial information. So we use another strategy called genetic algorithm to guide our optimization process. So basically, genetic algorithm is inspired by natural selection, and the key is how to select how we're going to define fitness in the setting. So our goal is trying to use the 20 genes to predict spatial information. So if you're going to select some genes that's the estimated XYZ coordinates are highly correlated with the gold standard coordinates, then we will consider that the selected genes have good performance. So thus, the fitness function in the GA algorithm that we defined is trying to maximize the mean correlation between the estimated XYZ using a method called multivariate random forest, which is used for multi-output machine learning method, and try to maximize the correlation between the estimate and gold standard XYZ for all of the cells. So this is a brief workflow of genetic algorithm. Due to time constraints, I'm only going to go over the basic steps and focuses on how we're going to assess fitness in our setting. So the first step is trying to initiate a first generation of gene sets, and each gene set contains 20 genes. And then we're going to assess the fitness based on what we defined before and select the top 5% fitness gene sets. And then using the top 5% fitness gene set, which is going to generate new gene sets by randomly exchange genes between the top fitness gene set in order to retain the good features in these gene sets. And then we're going to repeat this procedure again, and then until the fitness convergence. So how are we going to define fitness in our GA algorithm? So first, we're trying to estimate the spatial coordinates using the single cell RNA-seq data. So we use a method called multivariate random forest, which is a method developed in 2017 by Dr. Raman's group. So this method is based on decision trees. And what is different from this multivariate random forest and the conventional random forest is that it specializes in jointly estimating multi-outcome and takes into account of the correlation between XYZ coordinates when predicting. And its performance is better than independently predicting XYZ coordinates. So we use this in the training data set of single-cell RNA-seq data, and we use cross-validation to avoid overfitting. So the fitness function is defined after we get the estimated XYZ coordinates by using MRF. So for each cell, we're going to have XYZ's estimated by MRF. And then we also have the gold standard coordinates provided by the dream organizer. So we just want to see if the correlation between x-axis, y-axis, and z-axis are high. So that means that we already got gene sets that has high correlation and contains the most valuable information on spatial locations. I want to acknowledge that this is specific to this sub-challenge because we already know that there are gold standard coordinates here. It might not be generalizable to other settings, but it's specific to this challenge. So this is an overview of the genetic algorithm selection process. So we can see the x-axis is the generation of the GA, and then the y-axis is the fitness value defined in our setting. So the green dots represents that that's the best gene set in our generation. And you can see that at the beginning, the mean correlation between XYZ coordinates between estimated and gold standard is less than 0.8. And after 200 generation, the fitness reaching to convergence here is about 0.84 right now. So that's what we got as the final 20 gene sets in our GA selection. So after selecting the 20 genes, we use the similar mapping method as mentioned in sub-challenge 1 and 2. So we just use distribution map to get the top 30 genes and then delete the outlier and then choose the top 10 genes, top 10 position based on this map. So for evaluation, we also, as the first team mentioned, there are two scores that we can try to address. So the first one is the distance score that's already provided by the dream organizer. So we just calculate the average Euclidean distance between what we estimated and the true position by this map. And we also use the binarized data, and we didn't do any else on the binarized data. So for the gene pattern score, we use the selected gene expression correlation between the single-cell RNA-seq data and in situ data. We don't really know about the first scoring matrix, which is the genetic score, so we just use these two scores to guide our evaluation. So this is showing how our two methods performs for all of the three challenges. So the score that has a underline shows the best score among the two approach. So you can see that for the sub-challenge 1 and 2, we choose maximum distance because for sub-challenge 1, when selecting 60 genes, it has better distance score and the gene pattern score compared with the GA approach. And then for sub-challenge 2, we can see that the distance score here is better for MD method, but the gene pattern score is higher for the GA approach. So for sub-challenge 3, what we get is the GA approach get better distance score and the gene pattern score in this setting. So we choose GA for the third sub-challenge to submit, and then an MD approach for sub-challenge 1 and 2 to submit. So in summary, in using single-cell RNA-seq data, we use two methods, maximum distance and genetic algorithm method to select the most informative 60, 40, and 20 genes. And the selected genes has good performance on predicting spatial position and gene patterns. So I want to thank our mentors, Dr. Ke Xu and Dr. Ying Hu and Dr. Xinyu Zhang. Dr. Ke Xu and Ying Hu cannot be here because of schedule conflict, and all of them contribute a lot to support our analysis and provide guidance. And I also want to thank the dream organizer to providing a chance for us to present here. And I also want to thank everyone who stayed at the end of the talk and to listen to our presentation. Thank you.
Thank you very much. Several questions there. Let me go here first.
First of all, congratulations and a nice talk. I have a question about genetic algorithm that you have used with the cross-validation settings that you have. So I didn't understand which set of genes you choose because you are using different folds. So I was wondering if you can explain that step.
So for genetic algorithm, so first we generate randomly a population of gene sets. So from here, it's you can see it's kind of random at first, but there are some top performed fitness gene sets that we can find in this first generation. So we want to retain the genes that in the first generation to keep them. So that's why we select, let's see. So we select the top 5% of the gene sets for each generation. There are a lot of technical detail for GA, but basically it's starting from random, but keep the best feature of gene sets and retain them and try to make some randomness around it to try to see if there are new combinations that's available, and then try to repeat this process until it finds the best answer. It doesn't, I think the cross-validation is not affecting the selection process itself. It's how we're gonna estimate the coordinates. So I'm not sure if I can address this maybe in person in detail, but GA is kind of a complicated process.
So I think the question before wasn't about how the genetic algorithms evolves the solution. The question is, how can you take the coordinates X, Y, Z and run the genetic algorithm to find the best predictors, but then when you are going to do another fold, you are going to find another best 20 genes. And then at the end of the day, if you go and take the top ones that ranked across the different folds, you are already using the data from everything and you are overfitting. This is exactly the reason why we dropped our first solution and we tried to use an approach that would use cross-validation, but here cross-validation cannot be done to do feature selection. It's just not possible. While using the XYZ coordinates, you cannot do cross-validation.
I think I can answer your question. And you said GA is an aggressive method. But here we use two important steps to prevent overfitting. One, the most important is we use a different mapping method to evaluate it. Even though we choose the genes with GA and MRF, but we did mapping use MCC. MCC is a common traditional method. So if there is overfitting in GA step, MCC will not work well.
So you use MCC between the binarized drop seed and the in situ?
So I want to make it clear that when we try to estimate the coordinates, we didn't use any of the in situ data. We just used the RNA-seq data. So for mapping, it's the same as the first performer. We use MCC because it's the gold standard also use MCC. It's closer to their estimation.
Here. So as far as I understand, so the population is composed of randomly chosen sets of 20 genes, right? And then you have to apply some crossover and mutation operators. What kind of crossover and mutation operators you use? Because here you're working with numerical data.
You mean cross-validation
No.
amount?
Crossover operator for the
Oh,
genetic
crossover. Yeah, so we actually have a slide for that. Let's see if we can find it. So, let's see. Here. Oh. So actually, we just use the 20 genes, just like a chromosome. And for crossover,
I mean, specifically, do you use a single crossover, double crossover? Which operator do you use?
Oh,
I
it's
mean,
just a
there
single
are
crossover.
widely known operators for that.
yeah, it's a single
Single.
crossover.
And how about the
Right.
mutation is just randomly replacing a gene?
Yeah, so it's here. Like there might be some randomness created by mutation in the GA process.